Welcome everyone to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi and I'll be hosting today's session on the importance of chemical synthesis for antimicrobial R&D. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and are freely available to view after the live broadcast on our website, revive.gardp.org/webinars. On Revive, you can also read our series of articles known as Antimicrobial Viewpoints. Here, experts discuss various topics within the field. We also have a resources section that includes the Antimicrobial Encyclopedia. This has definitions of various important terms that you'll hear of in today's webinar. Some of these also include videos where experts give a further explanation on the terms. You can find one currently on Hit to Lead by Ravi, who will be moderating today's webinar. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar by clicking on the questions button as shown here. We will address the questions after the presentations and do our best to respond to as many questions as possible. If your question is for a specific speaker, please do indicate their name at the beginning of the question. Today's speakers are Anna Hirsch and Patrizio Mattei. Our moderator today is Ravindra Jumde. Ravindra Jumde is a senior scientific officer in the Discovery and Exploratory team at GARDP. He's an organic chemist with a solid background in drug design, optimization, and development. Ravindra previously worked at the Helmholtz Institute for Pharmaceutical Research Saarland, HIPS, where he led projects in drug design, contributing significantly to the development of inhibitors for various bacterial and viral enzymes. Ravindra received his PhD in chemical sciences from the University of Pisa, Italy, and has held several positions at the University of Pisa, ISTM CNR Milan, and the University of Groningen, Netherlands. Welcome, Ravindra. I now hand over to you to introduce our first speaker.
Thank you, Victor, and welcome all to today's webinar on the importance of chemical synthesis for antimicrobial research and development. Special thanks to the speakers of today's session who accepted our invitation to give us more insight into their work. Now it is my pleasure to introduce our first speaker, Professor Anna Hirsch. Anna is a full professor of pharmaceutical chemistry at Saarland University and a head of the Department of Drug Design and Optimization at Helmholtz Institute for Pharmaceutical Research, also known as HIPS. Her research focuses on anti-infective drug design, adopting rational approaches such as structure and fragment-based design in combination with the protein templated strategies of dynamic combinatorial chemistry and kinetic target-guided synthesis. She has published over 170 peer-reviewed articles and received several prestigious grants. Anna completed her PhD at ETH Zurich in the research group of Professor François Diderich. She then moved to her postdoctoral studies to the group of Jean-Marie Lane at the Institute of Supramolecular Science and Engineering in Strasbourg, France, before she joined as an assistant professor at the Stratting Institute of Chemistry at the University of Groningen. I am now happy to welcome Anna, and Anna, the floor is yours.
Yeah, thank you very much for the kind introduction, and I look forward to giving you some insights into the work that we conduct at the HIPS. And somehow the slides are not moving. There we go. All right, so there seems to be some delay when I click for the next slide, but let's see how that develops. So of course, all of you certainly know how difficult, lengthy and expensive the development of new antibiotics is. This is a situation that was recognized in Saarbrücken on the campus, where I still don't get to the next slide, where the local university, Saarland University, and the HIPS, the Helmholtz Institute that is on the university campus, decided to join forces, thereby taking advantage of excellent computer science and bioinformatics expertise on campus, paired with the medical faculty and of course the natural science faculty. And all players across all of these disciplines are really eager to engage in drug discovery. And this has recently been formalized in what we call the Pharma Science Hub, which is an unprecedented collaborative platform where we actually also have the chance to integrate industry on campus. You will certainly hear more about this in the coming months and years. Now, what exactly is the HIPS and the Helmholtz network in general? So the HIPS is actually an institute that is a satellite of the larger Helmholtz Center called the HZI. And at the HZI, which is located in Braunschweig, a different city in Germany, we deal with the topic of infection research from 3 angles, which are called topics 1, 2, and 3. Topic 3 is clearly the most relevant for today's webinar, and this is mainly being dealt with at the HIPS in Saarbrücken. We have different sites, but for today it's clearly the HIPS in Saarbrücken that is the most important. And we are also benefiting largely from the German Center for Infection Research, the DZIF, where we also had the translational research unit focusing on novel antibiotics. So to address the challenging task of finding new antibiotics or anti-infectives, we have in the recent years expanded our team, strengthening the natural product scientists and also the medicinal chemists. But currently, of course, it's getting more and more important to flank this with bioinformaticians and, of course, also colleagues that are eager to add in artificial intelligence approaches. And just in case the experimentalists don't manage to optimize the properties of a compound, we, of course, also have a drug delivery department. And a few of our colleagues are complementing our team in Saarbrücken, but they are based at the HZI in Braunschweig. At the HIPS, we tackle the task of finding new antibiotics by pursuing 2 research lines that at least initially start in parallel. One is looking for new natural products, be it from a bacteria, for example, a bacteria that live in the soil, or more recently also the microbiome. And of course, the complementary approach is to adopt a medicinal chemistry program where we focus on a certain target protein. Now, these lines often converge, and there's certainly almost always synergy and interaction. This could be that a target gets discovered through a mode of action study of a new natural product that could then be used in a medicinal chemistry program. Or it could be that a medicinal chemistry is needed to optimize the properties of a given natural product as a hit or lead compound. And because we are in the Helmholtz Association, we actually also have a mission to take promising projects further down the translational path and hopefully as close as possible and possibly even into the clinic. Now, at the Department for Drug Design and Optimization, we have pretty much a generic workflow where we are very keen to integrate computer-aided approaches and, as mentioned already, also artificial intelligence. This could be a simple structure-based virtual screening campaign. But we are equally eager to also look for experimental approaches to find new hit compounds. And this could be a fragment screening campaign or one of the protein templated strategies. In all cases, we heavily rely on biophysical assays to get a first confirmation on whether our hits are indeed binding to the target. Then a lot of synthesis follows, which usually gets us quick access to structure-activity relationships, especially when we're dealing with typical drug-like chemical classes, so synthetic small molecules. And it's important because we're dealing mainly with underexplored targets to then also establish the corresponding target-specific assays on the biochemical side, and flank this, of course, by whole cell screening, and as the project progresses, also by increasingly complex ex vivo and eventually in vivo models. Wherever we can, we of course love to take advantage of structural biology, be it X-ray crystallography or cryo-EM. And because I mentioned that we really want to take translation seriously, we are also eager to subject our compounds to pretty systematic in vitro ADMET screen profiling that then enables us to select the most promising compounds for the in vivo PK and PD studies in the most rational way possible. So I mentioned these protein templated approaches. This is maybe a more fundamental academic aspect, but it does sometimes deliver interesting hit compounds that could then well become the focus of a translational project. And these strategies come in two flavors. One is dynamic combinatorial chemistry, where we rely on a reversible covalent bond forming reaction. And the second related strategy is kinetic target guided synthesis, where it's actually an irreversible bond forming reaction. In both of these approaches, there are still a number of bottlenecks that need to be addressed. One being the rather limited list of chemical reactions that have shown to be compatible with these protein templated strategies. The size of the libraries of building blocks that one can use is certainly also a limiting factor, and it also remains to be demonstrated how broadly applicable these approaches are across different classes of proteins. And these are questions that we have been trying to address over the past years, combined with ideas where we could potentially combine these protein templated strategies with other hit identification strategies and benefit from synergy. One example being de novo structure-based drug design that can benefit from a combination with either approach. But let's now focus on our portfolio. These are some of the target proteins that we have selected to look at. As mentioned, they are often under or even unexplored, which we feel is important if we really want to find new anti-infectives with unprecedented modes of action. I will not bore you with all the details, but reasons for including a target protein in the portfolio could be that it has been not looked at yet. The ECF transporters are one example. Or they could be unique to pathogens, for example, the enzymes of the methyl phosphate pathway. Or there could be other good reasons, for example, them falling into the category of pathoblocker or antivirulence targets. Of these projects, a few have emerged as looking particularly promising, and in these cases, we are indeed trying to move them closer to the clinic, in all cases facilitated by dedicated funding instruments like CARB-X. So for today, I will give you just two examples. The first one deals with the target protein DNAN, and I will briefly look back in time and try to summarize where this project has come from. So grizzlymycin is a very old natural product, and my colleague Rolf Müller and his group tried to unravel the mode of action. And in this way, they got to this very interesting target protein, DNAN. Through a collaboration with Sanofi, they then generated close to 300 derivatives of this natural product, and it all looked very promising, and it looked like there could be a very nice future anti-tubercular agent. But this program had to be stopped because of a very unexpected liver toxicity issue in an advanced dog study. At the time, we could have decided, of course, to stop this program, but we decided not to do so because we feel it's a very interesting target. It's this ring-shaped processivity factor for the bacterial DNA polymerase that is essential for replication, and it's also highly conserved across bacteria and sufficiently distinct from the eukaryotic counterpart. In addition, the grizzly-mycins have a very low frequency of resistance, and the mutants suffer from high fitness costs. Now, because the existing starting points, the grizzly-mycins on the one hand, and the reported synthetic inhibitors were not good starting points, we decided to start from scratch. And at the moment, we are actually tackling this target from the two angles. So there is a natural product program with a new starting point called mycoplanacin. And here, this is still looking at TB and focusing on TB. And in addition, there is by now a new class of synthetic small molecule inhibitors that are showing a much broader antibacterial spectrum. And these are currently being developed as new agents for the treatment of community-acquired bacterial pneumonia. So when we have such a new target from a medicinal chemistry point of view, we usually try to spread our risks and explore different hit identification strategies in parallel. For today, I will focus on this structure-based virtual screening approach and briefly also summarize the dynamic combinatorial chemistry. So in the DCC approach, we took advantage of the ample structural information to inspire the design and selection of the building blocks and relied on acyl hydrazone formation as a reversible covalent bond forming reaction. In this way, we could generate the first dynamic combinatorial libraries where you essentially look out for amplification of some of these peaks in presence of your target protein. And these amplified library members then have to be resynthesized, and of course their binding to the target needs to be confirmed, which we did with SPR and STD NMR spectroscopy. And these DCC hits are looking very interesting. They have the most interesting activity still against mycobacteria, just like the natural products, but they are showing a broadened spectrum. And we have recently managed to achieve a breakthrough in hit optimization by being able to design out the isoniazid motif that was actually one of the building blocks. So the second approach that we used for DNA-N is structure-based virtual screening. And here a very conventional workflow using commercial libraries and two biophysical assays, SPR and MST, combined with phenotypic screening enabled us to validate 3 hits. One chemical class looked particularly promising because it's a new chemical entity that shows very good affinity and has a broader antibiotic activity spectrum with no cytotoxicity. So this encouraged us to look at these hits more closely, and we could show that they are indeed functional inhibitors in a DNA replication assay. And we could also show that there is a shift in MIC value when we take a grizzly mycin-resistant Mycobacterium smegmatis strain where DNA-N is amplified and overexpressed. Eventually, after a lot of work, we also found that Rickettsia typhi DNA-N is the only DNA-N homolog that is compatible with soaking, and that actually enabled us to get crystal structures of this new chemical class, which is of course an important starting point for a full-fledged hit-to-lead optimization campaign. And this is currently being funded by CARB-X, and we have established structure-activity relationships with, for the moment, around 200 compounds where we start to understand what is favorable in each of these moieties. I'm afraid I cannot show you the structure yet, but because of today's topic, I just want to ask you to trust me that this is a very interesting chemical class with a rather unique chemical scaffold. So we have now profiled these first inhibitors and have gathered more and more data where we can show that the antibiotic activity is improving, and we still don't see any cytotoxicity issues. And we also are very happy that the in vitro ADMET parameters are looking very favorable with high plasma and metabolic stabilities. The only parameter that we still have to optimize on is the plasma protein binding. So hopefully this will enable us to conclude the hit to lead optimization smoothly and then move on to the next stage. I will now switch gears and take you to the second case study where I have picked one of our pathoblocker targets. And just as a reminder, as opposed to a classical antibiotic that of course aims to kill the bacteria or delay their growth, which inevitably leads to the development of resistance, the pathoblockers are only aiming at disarming the bacteria. And because of this milder or perhaps even more pacifist approach, the expectation is that there's a lower selection towards resistance. And in addition, because they are addressing a pathogen-specific virulence factor, they should preserve the commensal bacteria. For us, Pseudomonas aeruginosa is the target pathogen, which is of course most important for infections of the lungs, but also the skin or wounds, eyes, and the urinary tract. This pathogen has as a key virulence factor the secreted metalloprotease called LAS-B. And this is a very interesting target because, of course, it's extracellular, so we don't have to enter the gram-negative bacteria, which is important when it comes to the properties of the classes of inhibitors. And it has important functions. It basically degrades the host's connective tissue and thereby helps the bacteria disseminate, and also degrades components of the host's immune system, helping the bacteria to evade the immune response. Furthermore, this target is validated in vivo and highly conserved across clinical isolates. So it's a very attractive target for the development of new pathoblockers targeting Pseudomonas aeruginosa. Now, this project started with a, sorry for the delay, with a functional screening of a protease inhibitor library that gave us access to this first very small fragment-like hit compound. And this project actually started before I joined the HIPS in the group of my predecessor, Rolf Hartmann, that was very kind to pass it on to me when I joined the HIPS. So from this hit compound, we learned from the first complex crystal structure that it's the free thiol that is the active inhibitor, and the thiol, of course, nicely complexes the zinc cation. And what was very interesting for us was that there was actually a second fragment hit bound close to the core of the active site in a subpocket. And this, of course, immediately inspired ideas of how we could grow, link, or merge these fragments. And because of this wonderful structural basis, these attempts were very efficient, and with only 55 derivatives, a rather small set, we could quickly take our inhibitors into the nanomolar range by identifying this alpha position as the best growth vector. These compounds are highly selective over a panel of human off targets. These are all zinc-dependent enzymes, and they also don't show any cytotoxicity. And in this program, as opposed to DNA-N, we are very fortunate that we get access to high-resolution crystal structures very rapidly, sort of on time for the next round of design and synthesis. Now, with this free thiol in hand, we convinced CARB-X to take it into their portfolio and start a hit-to-lead optimization campaign, where the first task was clearly to get rid of the free thiol group. So a systematic screening of alternative zinc binding groups identified the phosphonate as a very attractive moiety because it leads to chemically and metabolically perfectly stable inhibitors that are in addition also more active and are very soluble. So with the structural support, we could quickly grow this fragment further in the pocket and get to the first single-digit nanomolar inhibitor. And again, because of this wonderful structural biology support, we could also expand and diversify our initial class by taking us into 5 different chemical classes. They are all looking extremely interesting. They're highly potent, selective over the human off-target panel, don't interfere with standard of care antibiotics that would have to be co-administered, and their very favorable in vitro ADMET parameters nicely translate into in vivo PK profiles. So we have a very good position to plan the first in vivo PD experiments. And as a first quality check, we like to use Galeria melanella larvae as a first simple in vivo model. Sorry, the animations are not responding always equally quickly. And here we could immediately see that our LAS-B inhibitor improves the survival rate of the larvae. So this then meant that we could go further and again think about the possible target indications and target product profiles. So when we look at the lung, we can either think of acute pneumonia, hospital-acquired or ventilator-associated pneumonia, where IV would be the route of administration. And if we think about cystic fibrosis or even non-cystic fibrosis bronchiectasis, inhalation would be the preferred route of administration. For the eyes, clearly it would be a topical route of administration. And I'm very happy to share with you today that we have in the meantime achieved in vivo proof of concept in the murine infection model for all 3 routes of administration. And this we feel is also thanks to the presence of the phosphonate that leads to very good PK profiles, almost disconnected from the route of administration that we consider. I will just back this up with data from 2 of these in vivo studies. The first is for the inhalative setting, where we administered the LAS-B inhibitor in combination with levofloxacin as a standard of care antibiotic and saw a significant reduction in CFU. And to corroborate that this is indeed coming from inhibition of LAS-B, we also looked at the LAS-B levels in the blood. And here we see that they're significantly reduced both in combination and also after the single administration of the LAS-B inhibitor, which is a very nice way of confirming target engagement. For the second possible indication, Pseudomonas keratitis, we have teamed up with the local medical microbiologist Markus Bischoff at the medical faculty. And in the keratitis model that his group established, they could firstly see that the combination leads to improved clinical scores. It also leads to a reduction of CFU, again, especially in the case of the combination treatment. And to gain more confidence, we again looked at additional biomarkers. And here we saw, for example, that the immune cells in the eye were reduced after the combination treatment. We also saw that there were no major effects on the immune cells in the blood. And finally, we also looked at inflammation markers and found that IL-1 beta levels were significantly reduced after the combination treatment. So, and this is of course all looking very promising, and we really hope that we will be able to continue on this promising target with these different chemical classes. They look all so promising at the moment, but we are starting to see that some classes might lend themselves better to one indication rather than the other. This has all been possible because of extensive funding that we've managed to acquire that enabled us to accomplish hit-to-lead optimization and also make good progress in lead optimization. We are probably very close to the end of lead optimization and hope that this will be possible in the near future, and you will certainly hear about this. So with this, I would like to finish my talk and just give you a few take-home message, namely that target-based anti-infective drug discovery can be very successful. We like to spread our risks and consider multiple identification strategies in parallel, and are very keen on including multi-parameter optimization from the earliest possible stage. And with this, I would like to thank all the people involved in my group, the many collaboration partners, and also the funding agencies, and you for your kind attention. Thank you.
Thank you very much, Anna, for a really nice and clear talk. So now we move next to the next speaker, and our next speaker is Dr. Patrizio Mattei. Dr. Patrizio Mattei is a distinguished scientist in medicinal chemistry at Roche Innovation Center, Basel. His research interests have been mainly devoted to the areas of metabolic diseases, ophthalmology, and more recently, antibiotics. Patrizio led the medicinal chemistry team that discovers the novel antibiotic everyone knows as zosurabalpin. So before joining Roche in 1999, Patrizio received his doctoral degree from ETH Zurich and was a postdoctoral research fellow at Scripps Research Institute in La Jolla, California. Welcome, Patrizio, and you may start your presentation now. Looking forward.
Yeah, thank you very much, Ravi, for a kind introduction. Let's see how this. Yeah, I'm delighted to present to you our case study on chemical synthesis in antimicrobial R&D. I will introduce zosurabalpin, the first representative of a fundamentally new class of antibiotics, which selectively targets Acinetobacter baumannii. Zosurabalpin is a synthetic antibiotic, therefore chemical synthesis has played a central role in this project. And as you will see, the synthetic foundation of zosurabalpin has significantly evolved from one project stage to the next. Acinetobacter baumannii is a species of gram-negative bacteria which has been known for more than 100 years. Infections caused by Acinetobacter were rare and could be treated by a range of antibiotics as recently as 20 years ago. The situation has changed dramatically since then, and now the majority of Acinetobacter infections are caused by carbapenem-resistant strains against which our common antibiotics are ineffective. The World Health Organization has declared carbapenem-resistant Acinetobacter a #1 priority pathogen for which new medicines are urgently needed. The carbapenems have long been the treatment of choice for Acinetobacter infections, but they are obviously no longer working against carbapenem-resistant strains. The current standard of care represents a combination of drugs which act through established modes of action, and Acinetobacter already starts to be increasingly resistant against these drug combinations. At Roche, our vision is to significantly improve treatment for Acinetobacter by resetting the clock of resistance. To achieve this, we are introducing new classes of antibiotics which kill bacteria through previously undrugged mechanisms of action. But where do we start? For decades, the pharmaceutical industry has been screening their corporate libraries in order to find new starting points for antibiotics, unfortunately with limited success. Therefore, we looked out for cool molecules which we haven't screened yet. Transzyme Pharma has presented an attractive class of molecules. Their hit-create library is composed of tethered macrocyclic peptides, which also go by the abbreviation of MCPs. The MCPs contain a tripeptide part and a non-peptidic tether to close the ring. This arrangement offers novelty, structural diversity, and small molecule drug-like properties. We got in contact with Transzyme when they showed us the results of a phenotypic screen for bacterial growth. There was one HDS hit that really stood out. The molecule features an M-methyltryptophan subunit, which is an essential motif for this class. Remarkably, this molecule was selective for Acinetobacter and was not active against other bacteria. In a few rounds of optimization, we arrived at our first-generation lead. Substitution of the central ornithine by a lysine, as well as the 2 chlorine atoms at the southeastern ring improved the potency, while the pyridine nitrogen reduced the amphiphilicity and dialed out off-target activities. We were delighted to see that our lead compound worked in vivo, but unfortunately the compound caused plasma precipitation after intravenous infusion as well as organ toxicity. Such a compound was obviously not developable. Through exemplary collaboration between chemists, biologists, formulation scientists, pharmacokineticists, and toxicologists, we were able to establish a new generation of zwitterionic MCPs, which were causing much less plasma precipitation and caused no organ toxicity at therapeutic doses. Of these compounds, zosurabalpin was selected as a development compound, which is currently in phase 1. Here is the efficacy of zosurabalpin in a lung infection model in mice. In this experiment, the lung infection is caused by a clinical isolate against which there is no efficacious drug at all, and sadly this has become more and more the reality in hospitals. In such situations, zosurabalpin addresses an unmet need and could save lives. The unique efficacy of zosurabalpin in comparison to established antibiotics is indicative of a new mechanism of action, which I will get to later. On the right panel, you see a snapshot of human pharmacokinetic data from the phase 1 clinical studies. Here, a 600 mg dose was infused intravenously into healthy participants and later into intensive care unit patients. Gratifyingly, zosurabalpin is well tolerated both in healthy participants and patients, while exposures in patients are typically higher and have greater variability. In collaboration with Harvard University, we uncovered the mechanism of action of zosurabalpin. Here you can see the cryo-EM structure of zosurabalpin within the lipopolysaccharide or LPS transporter. Surprisingly, zosurabalpin was found to form a complex with a transporter and a bound LPS molecule, which is a unique mode of inhibition. This structure and a number of additional experiments described in the paper below helps us understand how zosurabalpin works. Let's first look at the uninhibited LPS transport system in Acinetobacter. The inner membrane transporter LPTB2-FGC picks up LPS, and after closure of the lateral LPTC helix gate, LPS is transported to its final destination at the outer membrane via a bridge-like arrangement whereby the transporter consumes one equivalent of ATP. Now add zosurabalpin to the equation and look what happens. Zosurabalpin is the red diamond shown here. It enters the cell and binds both to the inner membrane transporter and LPS, thereby trapping the LPS molecule within the transporter and blocking further transport. The lateral gate cannot close, but the transporter consumes ATP even at a higher rate. This process leads to Acinetobacter cell death. Finally, the molecule is only hitting Acinetobacter and none of the other gram-negative bacteria, and this is due to structural differences of the LPS transporter binding site. As I mentioned at the beginning of the talk, zosurabalpin is a fully synthetic molecule. It is not derived from a natural product, nor does it mimic the natural substrate LPS. Obviously, you need chemists to produce these molecules. The first relevant synthesis was the synthesis of the screening library, which contained the starting point of zosurabalpin, but was also used in other projects. Chemists at Transzyme made all these molecules in milligram quantities. Once the hit was identified, medicinal chemists at Roche produced hundreds of new drug candidates, first in lead identification, then in lead optimization, until we arrived at zosurabalpin, which we believe could become an antibiotic for patients. At clinical candidate selection, the role of chemical synthesis changed fundamentally. From that moment on, the goal was to develop the best possible synthesis of the zosurabalpin, and only of the zosurabalpin, to supply kilogram amounts of material for clinical studies and ultimately for patients. So we are dealing not with one chemical synthesis, but with several, and at each project stage different considerations apply. The synthesis of the HDS library has to be very versatile and produce a great variety of target compounds rapidly from readily available building blocks. In lead identification, the attention shifts to chemical diversity. The systematic exploration around a given chemical scaffold often requires significant modifications of the initial synthesis. Purification becomes more relevant. In lead optimization, we typically have a pretty good idea of where we want to go. The chemical diversity decreases and focuses on critical part of the molecule. Because we are considering several drug candidates in parallel, the synthetic strategy often involves key common intermediate for late-stage functionalization. At the same time, synthetic complexity can increase when fine-tuning of the lead compound results in unusual substituent design. The best compounds require synthesis in larger amounts for animal studies, hundreds of milligrams for efficacy studies, a few grams for the first exploratory tox studies. After clinical candidate selection, the focus of the synthesis shifts entirely from chemical diversity to the best possible synthesis of exactly one compound. In drug development, much larger amounts of material have to be delivered under time pressure. At the same time, the reagents and solvents, as well as the empiric characterization, have to meet regulatory standards. Once the drug is on the market, the synthetic process has to ensure the supply to patients while being cost-effective and robust. Therefore, each one of these stages has its own synthesis challenges requiring different chemistry skills, and to meet these challenges, different chemists are in charge of the synthesis at any project stage, from library synthesis experts to medicinal chemists, process and manufacturing chemists. The synthesis of the HDS library is a very elegant case of solid phase synthesis on a derivatized aminomethyl polystyrene resin. The Transzyme chemists have used the DDZ protective groups, which can be removed under conditions that leave other acid labile groups like T-butyl carbamates and T-butyl esters intact. The benzothiazole sulfonyl group, on the other hand, acidifies the N-terminal amine, enables a methanol reaction with the alcohol of the tether here, and can be easily removed after macrocyclization using polystyrene-bound thiophenol. Macrocyclization also cleaves the products of the resin, and finally the protective groups of the amino acid side chains are removed. This library was very diverse in composition of the tripeptide subunit, but less so in the non-peptidic part, as it was limited to off-the-shelf starting material. Here is the commercially available tether of the HDS hit, and you cannot really buy anything similar, not even the diaryl ether analog. But the main limitation of the synthesis was the stability of the thioester attachment to the resin, which made the approach unscalable because the peptide fragment was unable to fully hold onto the resin over all the steps. Now let's jump to the discovery synthesis of zosurabalpin. We are now using the 2-chlorotriethyl resin for attachment of the C-terminus to the peptide, which is a much more robust linkage enabling standard FMOC chemistry. The tripeptide and the tether part are unified in a reductive amination reaction, Then the FMOC group is removed and the peptide is cleaved off the resin, and macrocyclization is the next step. In lead optimization, fine-tuning at this position using the late-stage intermediate here has enabled us to identify the clinical candidate zosurabalpin, and this synthesis could be scaled up to gram amount. In lead optimization, a lot of effort was spent in the synthesis of the tether molecules. Even though the change from the original tether was minimal, there is just a pyridine nitrogen and a bromine. These changes entailed the change from an off-the-shelf building block to a 7-step synthesis. As you can see here, the 2 aromatic rings are brought together in an SNR reaction between thionicotinic acid and 2-bromo-6-fluorobenzaldehyde. After esterification, the aldehyde is derivatized by a reaction with T-butyl sulfonamide. Then both functional groups are reduced and the sulfonyl protective group is removed by hydrogen chloride. Finally, the amino group is FMOC protected and the alcohol is oxidized to the aldehyde. The shortcoming of the synthesis was the cleavage step of the resin, which had to be carried out in a few minutes in order to minimize epimerization. Although this is practicable on a relatively small scale, it is not possible to scale up to the amounts that are required to run GLP tox studies, let alone clinical studies. The discovery synthesis also had other minor issues like multiple chromatographies, the use of trifluoroacetic acid, and a hygroscopic amorphous product with poor solid-state properties, but that's really quite the rule at this stage of the project. Now fast forward to the synthesis that our process chemists worked out. This is now a scalable synthesis, and the key to making large amount is the isolation of crystalline intermediates like the tripeptide hemiphosphate and the 2 macrocyclic intermediates, which greatly simplify the purification. This is really important because the increase of scale is considerable, from a few grams to hundreds of grams and then kilograms in the clinic, and the time pressure is significant. Also, zosurabalpin itself is a crystalline product, which again helps scalability. In more detailed, solution chemistry really has been the solution to suppress epimerization, and the resin has been replaced by an allyl ester. The seemingly trivial amide coupling step is arguably the key step in a synthetic route, and these reagents were among the few under which epimerization at the lysine stereocenter was minimal. In the final deprotection step, hydrochloric acid could replace the previous procedure with trifluoroacetic acid. Zosurabalpin is a remarkably stable molecule which stays intact even in concentrated hydrochloric acid solution at 50 degrees. I suppose that we were just not brave enough to try these conditions in discovery. Zosurabalpin is isolated as the parent molecule, which is non-hygroscopic and has well-defined and reproducible solid-state properties. The scalable synthesis is not only concerned with the product, but also with the byproducts, impurity from reagents, and residual solvents. For instance, the Suzuki coupling reaction requires a palladium catalyst late in the synthesis, and there is a strict regulatory limit for palladium in the final product of just a few ppm. An example for a nasty side product is dibenzofulvene, which is formed during the deprotection of the FMOC group and is difficult to remove if the desired product, like this one, is not crystalline. Therefore, the process chemists developed the opposite approach. Here, dibenzofulvene is scavenged with N-acetylcysteine, and this adduct is removed by precipitation of the poorly soluble dihexylamine salt. From a medicinal chemist's perspective, this synthesis of zosurabalpin is impressive and represents a huge improvement over the original route. This route has been used to produce zosurabalpin in multi-kilogram amounts needed in drug development. However, from a commercial point of view, this route is still not ideal for several reasons. For instance, the procedure for FMOC removal is certainly not atom economic and involves more handling steps that one would like. So a route not involving FMOC chemistry at this position would be desirable. Then we have hazardous reagents like hydroxybenzotriazole and hydroxyazobenzotriazole, which is unwelcome and can entail complicated processes to ensure adequate safety. Therefore, an ideal commercial route should guarantee the supply to patients in a cost-effective manner. The process has to be robust and safe and should only use unproblematic reagents. Our chemists in process development have already made significant progress, and they will present an improved and commercially viable synthesis of zosurabalpin already next week at the Swiss Industrial Chemistry Symposium in Basel. And this brings me to the summary of my presentation. I have introduced zosurabalpin to you, a novel antibiotic to treat difficult-to-treat Acinetobacter baumannii infections. Chemists have played a fundamental role in the discovery of zosurabalpin, first by making the screening library of 45,000 new molecules, then by synthesizing several hundred more new molecules through an iterative optimization process. My final take-home message for you is that in a drug discovery project, the role of chemical synthesis undergoes a fundamental change at clinical candidate selection. Early project stages up to lead optimization are chemical diversity-oriented processes. Medicinal chemists invent and produce several new drug candidates, but after clinical candidate selection, the task of chemical synthesis is handed over to process chemists, who create the synthesis of the selected molecule to supply the product required for drug development, with the potential to be later technically developed into a large-scale production process. Finally, I would like to thank the project team for their dedication and tenacity, especially our chemistry heroes. Konrad Bleicher, our peptide chemist in the early phase of the project, Jean-Michel Adon, our first process chemist, Fabien Hoffmann-Emery, and Ernesto Santandrea, who with their teams have brought the scalable synthesis of zosurabalpin to its current state. I also wish to thank our external collaborators and BARDA for funding. Thank you for listening, and I'm happy to take your questions.
Thank you very much, Patrizio. It was really interesting journey. How do you develop the zosurabalpin? So, we will now start the question answer session. So, quick reminder for all the audience: you can submit your question as shown on this slide. We will do our best to respond to many questions as possible during the allocated time. So, let's start with some common questions, where I would like to get maybe opinion from the academic expert like Anna and the industrial expert, their opinion on this kind of questions. Like the first one was, what about the risk of development of resistance in the pathogens against this new class of chemical antibiotics in long run? So either of you can start here.
Maybe I can start by saying, of course, that is, resistance will always develop. It's always a question of time. And this might be connected to the targets. So there are some targets for which the frequency of resistance development appears to be lower. This non-traditional strategy of the pathoblockers promises to have in general a much lower frequency of resistance, but this has to be demonstrated experimentally. So this has not been done by any group in the field yet, and we are working on this, but it's actually not that easy to do this experimentally.
Yeah, what I can add to this is that, as Anna said, resistance will develop inevitably. Bacteria will always outsmart us in the long run. What we can do or what prescribing doctors can do is to prescribe antibiotics responsibly. So, for instance, not prescribe them for viral infections, and really use them for their intended indication. Then diagnostics are uh, developing rapidly. So in the long run, we hope that we can actually diagnose the type of infection that a patient has. So if you know that the infection is caused by a particular species, I don't know, like E. coli or Acinetobacter, you will give the patient uh, the antibiotic that effectively targets that kind of infection, and not something that just tries to kill everything.
Good. Yeah, thank you. So the second common question is, yeah, I think natural products versus small molecules. What are your takes? So which kind of research area could could lead to a new class of antibiotics, and also taking into account the total cost of developing this kind of drugs and also the resistance development afterwards?
Patrizio, do you want to start?
I can start. I mean, historically, the majority of antibiotics are either natural products or derivatives of natural products, but there are also successful examples of fully synthetic antibiotics, like the quinolones or the oxazolidinones. So, and I don't know. I mean, zosurabalpin is a fully synthetic product, but it looks a bit like a natural product because it has amino acids in it. So I don't think it's helpful to make that distinction between natural products and non-natural products. I think the whatever molecule does the job is appropriate, and we should be open-minded and not exclude uh, any chemical class a priori just because we haven't seen it yet. Because antibiotics are known to be sometimes a bit weird-looking molecules. So yeah, let's stay open-minded.
Yeah,
Nana?
so I fully agree. I mean, given the status of the current pipeline, yeah, is it really still a pipeline? Is the first question. Really, I think must mean that we have to be open-minded. I couldn't agree more. So whatever it is, be it a very exotic natural product or a very boring synthetic small molecule, we should pursue. I mean, as mentioned in my introduction, at the HIPS, we are convinced that having both in parallel is definitely a good idea. It might not always be the natural product that would become the front runner, but it would often be a very good tool to get to a new target or to just get to a starting point for a follow-up medchem program. So hopefully also the natural product research industry might be resurrected. Otherwise, the academic groups have to keep it up.
Great. Yeah. Thank you for your answers. Uh, one of the question is for Patrizio. Have you been thinking about applying chemoenzymatic steps into the zosurabalpin synthesis? Its peptidic structure looks privileged for such approach.
Well, we haven't really been using any uh, let's say, any biology-inspired synthetic method, because you have to appreciate that it's only a tripeptide. So for us, it's rather a big small molecule which happens to have a peptide substructure. But yeah, I think with the the synthetic evolution from solid phase to solution phase has been quite straightforward, and because the building blocks were relatively standard, and you could basically you can buy the chiral center, yeah, there was not really a need to have more bio-inspired methods.
So yeah, one of the questions related to that. So you mentioned that. So you got this library from Tranzyme, right? Tranzyme library. Are there other microcyclic libraries available to test that you know, or?
Well, there are macrocyclic libraries. So at the moment, it's actually a company called Spirochem that commercializes like a version 2.0 of the original Tranzyme library. So now they have reduced the size of the library, but at the same time improve the quality of every single compound, so they can really stand behind the purity of each one of the members and can easily resynthesize them. So this is let's say the evolution of this library has gone very much into quality. But there are other macrocyclic libraries. There are, for instance, mRNA display, which is a very powerful method to identify promising starting points from huge libraries and do an evolution. Which is perhaps more a classical peptide chemistry approach.
Yeah. So yeah, related to that question about libraries, did you screen other classic small molecular libraries in this program? Like, or was really focused on this Tranzyme one?
In this project, we didn't, because we started the collaboration when Tranzyme already had done the screening, and because the screening was purely phenotypic, and it was only after the selection of the clinical candidates that we were able to identify the biological target and the mode of action.
Thank you. The next question is for Anna. It's regarding the phosphonate-based pathoblockers. So someone asked, does it inhibit ACE2, and is the potency affected by adding external zinc in the assay?
Yeah, so many thanks for this question. So we have this current off-target panel, which might be bigger. The way we actually put this together was by trying to cover all types of human zinc-dependent enzymes. And so far, we feel it's encouraging that we don't see any inhibition. And actually, something I didn't mention today is that these compounds were also subjected to a SEREP panel, the 44 safety panel, where we also don't see any flags or any significant flags at a concentration as high as 100 micromolar. The effect of additional zinc, no, this has not been a problem so far, but we are going to look into like maybe dissecting what contribution does the complexation of zinc have, and what does the rest of the scaffold have as soon as our new ITC arrives. The old one was unfortunately not operational for quite some time.
Right, yeah. And yeah, concerning about this last B, have we checked the activity of other zinc-containing targets in the bacteria, such as LPXC, while the head group may be a little large compared to the hydroxamic acid or LPXC enzymes, may be able to accommodate it?
So this is also a very good question. So we have looked at this in both directions. So testing reported LPXC inhibitors on LAS-B and the other way around. And we do see in some cases a dual inhibition, but it's rarely the case that the SAR is exactly running in parallel. And this is something that we have started to publish on, and the same holds true for another bacterial metalloprotease, the collagenase from Clostridium histolyticum, where we also see maybe some initial signs of a dual activity, But when you then really zoom in, you do see a divergence and like a preference of some motifs for one rather than the other target.
Thank you. Coming back to Patrizio about this 40K library which you purified. So it seems like it was not clear maybe for us. Like, are these 40,000 compounds pure enough after removal from the resin?
Well, they were pure enough to be useful in an HDS, but yeah, the sobering truth is that they are not really pure from, let's say, by organic chemistry standards. So I wouldn't consider them to be NMR pure. But
Mhm.
as Pyrochem has shown more recently, the, if you invest in purification, you can get them pure. But at that stage, when the screening was done, the purity was perhaps 60 or 70%, and the desired compound was the main component of what was in the vial.
Mhm. Yeah, so you see the activity and then you investigate like, okay, this is the major compound, and then purify the compound and then test it. And if the
No,
activity persists, that means that it's from the same
No, basically you have to resynthesize the
resynthesizer.
molecule because typically what uh, what was in the screening is such a tiny amount that you cannot really do much more with it. So you first resynthesize the molecule and then you get it pure and characterize it. And fortunately, in most cases, we could actually confirm that the compound was really what we thought it was.
Mhm. Yeah. And yeah, there was one more question about these hundreds of analogs which you made for the hit ID and optimization stage. How many changes of this tripeptide versus the tether you make? Like, so are there like mainly tripeptide changes, or there was like a combination between both of them?
Oh, of course. I mean, as you can see, the zosurabalpin has a change both in the tripeptide and in the tether. But initially, also the way the chemistry was set up, the medicinal chemistry was focusing a lot on the peptide part. And as I also showed, the synthesis of the tether even introduced uh, um, a really small change just entails a massive synthesis from this one step to 7 steps or even more. So this most of the most of the tether variations were done in lead optimization, and there we also benefited, or we strategically placed uh, 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 these intermediates that could be functionalized after macrocyclization.
So the late stage functionalization. So you had like an intermediate which can be used for, yeah.
Yes, yes. And unfortunately, not all the chemistry that you can find in the textbook works because uh, those are sterically quite demanding molecules. And for instance, Buchwald chemistry is really a nightmare because they usually require also huge catalysts, and they're just too big for the macrocycle.
And just yeah, remind me on that. So the final, the Suzuki coupling step,
Yeah.
is your like a huge molecule which can also act like a chelator to the metals? Would that be a problem for your Suzuki coupling step there, or was fine? Okay.
For the Suzuki coupling, it wasn't so, which also includes the dialyl sulfide. So uh, one may look at the sulfur and dislike it for some reason, but there was so it's not metabolized, it's not toxic, so there is nothing wrong with the sulfur.
Uh, for Anna, I have one more question for you from the chat. Was about, you mentioned optimization of plasma protein binding in one of your program. Can you elaborate on your thoughts in what way plasma protein binding is important in the context of antibiotics? What strategies do you employ to modify it in the desired direction?
All right. So I would say in general, it's important to find the right balance between all of these different parameters. And of course, the same holds true for plasma protein binding. It might well depend on, let's say, the route of administration and the target organ that you want to reach, what percentage would be beneficial. So, for example, in the LAS-B program, we feel that we might have to be in a given range to reach very high lung levels if we want to target the lung. But the question, I think, refers to the DNAN program where I mentioned that plasma protein binding is an optimization goal. So this is at the moment very high. We started off with 99.9%. And are now down to 97%. So making progress. We still don't know what we need to reach, but our hope would be that by having something that is not as high as 99.9%, we would actually reach higher levels after the administration. So we are at the moment conducting in vivo PK studies in a cassette format so that we can get a better feel for what might be required. And if we're lucky, we might get into a situation that we had or still have in the LAS-B project where we can actually use the in vitro data to sort of, in a rational way, decide which compounds would be most promising for the in vivo studies. The strategy to optimize it is to essentially make the compounds less hydrophobic. And this is a bit delicate because the binding pocket that we're targeting is very hydrophobic, and the natural products are also very hydrophobic. So it will be a very delicate balance.
Yeah, and related to that plasma protein binding, one of the question is like, did you perform these studies in a serum-free medium?
That's probably referring to the bacterial growth assays.
Bacterial growth, I think so. Is where the toxicity studies for compounds with high plasma protein binding performed in serum-free medium or not?
Yeah, that's an excellent question and a very current concern. So the cytotoxicity assay is a standard protocol where serum is present, and this might indeed mask the cytotoxicity. So we wouldn't trust it blindly in this way. But as a second assay, we are also looking at Candida albicans, and there the assay doesn't have any serum, and we now have compounds that have a very nice selectivity window, and so this is very encouraging for us and might well suggest that there is indeed no toxicity concern.
Yeah, and I think it's one of the question is for the LAS-B. So you mentioned that you tried different zinc binding groups. So other than the phosphonates, are you able to show a similar activity and ADME profile by other zinc binding groups, or the phosphonate was the best one?
So the person that asked this question, you can actually read about all the details in the paper that I cited. It's an ACS Central Science paper. Now there was actually more than one zinc binding group that looked promising. Besides the phosphonates, the hydroxamic acids also looked very promising. We decided to prioritize the phosphonates because of the, let's call it, bad reputation of hydroxamic acids. But in fact, all the hydroxamic acids that we've looked so far as LAS-B inhibitors have actually looked surprisingly selective.
Thank you. And coming to Patrizio, so there is a question about what is the predicted estimated cost for optimizing manufacturing cost per kilogram per dose of the zosurabalpin? Do you have these studies done? And what volume of the zosurabalpin do you anticipate needing for the manufacturing?
Yeah, I cannot really talk on that subject, but you will hear more about it in the near future.
Yeah,
Yeah.
can imagine. Yeah, yeah. Yeah, and yeah, the question about zosurabalpin during this whole process, did the knowledge of target and or structure-based design help medicinal chemistry process in your case, or?
Well, during the zosurabalpin project, we didn't know what the target was, so the optimization was purely phenotypical. So had we known the targets, there were many things that we wouldn't have done. But
Mhm.
yeah, it's a really trial and error, and you have to find out the hard way or at which positions can be modified or where substituents are tolerated. But it's a bit like a CNS project in the old days, so you'll figure out in some way. But of course, if you have the luxury of a crystal structure, that is of course very
Yes,
helpful. And when we had the crystal structure or the cryo-EM structure of the zorobalpin and the target, it was quite an eye opener because we were quite sure about the target before we had the cryo-EM structure, but let's say the you have to appreciate that this is a complex with four protein, two small molecules, and this complex with the zorobalpin and the LPS, how that might fit together was just beyond our imagination.
yeah, it's really. And so, oh yeah, about the administration route, do you have it in mind? It will it be IV or the oral or?
Yeah, so zosurabalpin will make the patient population are really the intensive care unit patients,
Mhm.
and for these patients, the established route of administration is intravenous, and because they usually cannot are not even able to swallow, so apart from the fact that zosurabalpin is not orally bioavailable, so it would not even be a plus.
Yeah. Yeah, and that brings me to the question to Anna as well about this LAS-B inhibitors. So it was clear from your talk they were either as inhaled drugs, but or IV. But did you try in your program to make them orally bioavailable, or is that an option at all?
So we have not looked at this yet explicitly because, of course, we don't have a target indication that would call for an orally bioavailable drug. But from the phosphonate and all the properties we know, and of course also from the PK investigations, we know that these compounds would be very amenable with an oral administration.
Mhm. Great. Yeah. And about this DNAN program, so we see that there is an activity against E. coli. So was that in wild type activity against the wild type strains? Or?
Yeah, so we have in our primary screening panel, we have the wild type strain and an efflux deficient strain. And so let's say at the beginning, we didn't see any convincing activity in the wild type strain, but in the course of the first round of optimization, this started to appear and is now looking more and more convincing. Um, for our target product profile, the anti-gram negative activity is not an essential requirement, but would be beneficial. So we will keep monitoring this and are very happy to see it already appear at this early stage.
Mhm. Yeah, so you already tried different gram-negative bacteria and tested these compounds on this WHO priority pathogen list, right?
Right, but we are within the CARB-X program, we are really focusing on the pathogens that are most relevant for the community-acquired pneumonia.
Okay, good. And for these DNAN molecules, are they so these are pure synthetic compounds, small molecules, or are they kind of macrocycles there? And and how do you tackle the off-target activity which is associated with the gristlemycin, right? So in gristlemycin, you see that liver toxicity, and do you have like a kind of structure-based design just to tackle this kind of off-target activity with these small molecules?
Okay. So the class that I focused on today is a classical synthetic molecule. It doesn't have any connection to the natural product. And we are pretty sure that we understand the underlying reason for this liver toxicity issue, which is inhibition of the bile acid transporter. So we can test for this in vitro, so it really appears to be limited to the natural product, to this macrocyclic scaffold. And nice thing about this new natural product class that I briefly mentioned, the mycoplanacins, is that they are not only orders of magnitude more active from the start, but they actually also show an even better selectivity than the most optimized gristlemycin derivatives. So this is really looking like a much more promising starting point. And this is remarkable because the structural differences between the gristlemycins and the mycoplanacins are minor, very minor.
Okay, good. Good. Yeah. And yeah, about the pathoblocker, there was a question. So what is the future of this kind of pathoblockers as a marketed drugs? Will that be kind of your view on that? Because they will be co-administered. And what do you think about that?
All right. So there are in principle two possible application scenarios. One would be the therapeutic setting, where I think everybody in the field agrees that co-administration and adjunctive treatment is the way to go. So we have actually 4 pathoblocker programs at the HZI, the 3 Pseudomonas programs at the HIPS, and 1 Staphylococcus aureus program at HZI in Braunschweig. And in some of the cases, we also see good activity for the monotherapy. And this in general would actually look very interesting for prophylactic application. And so this is something that might well be the easier path forward down the line, because you wouldn't have to demonstrate superiority. So this is currently actually the strategy we're pursuing. And in the case of LAS-B, we are also zooming in more on the eye infections, because we are also feeling that this might be the easier path forward when it comes also to the regulatory steps. And we would then sort of keep the lung infections not only in the back of our mind, but they could follow as soon as we have made good progress with the eye infection TPP.
Good, thank you. Coming back to Patrizio, so in case of this, was there any final product crystallization issue where their polymorphism issues were observed during that crystallization study?
Yeah, I mean, at the discovery stage, zosurabalpin was amorphous, and it was only later in development that our solid state expert actually discovered the crystalline forms. But I really don't know all the details about what was discovered and what wasn't. But what you see on the title slides are really zosurabalpin crystals.
Yeah, zosurabalpin crystals. So it was like then a salt form which was crystalline afterwards, like.
Yes. Yeah, the parent is crystalline. Yeah.
Great. Yeah, and coming to Anna about the co-crystal structures, in case in both of your program, at least in the last B, you were fortunate that you can get the co-crystal structures quite easily. And you mentioned sometimes about this DNAN. You had some issues with the co-crystallization. What was it, because of the target itself, or was this based on the molecule, also the class of compounds which you are now working on?
So the good news is that this target is highly conserved, and partially in collaboration with the Structural Genomics Consortium, by now a lot of structures of the homologs, of like all kinds of bacterial homologs, are solved, and they virtually look identical. But the subtle difference about this Rickettsia typhi homolog is that the pocket we're targeting is actually accessible within the crystal lattice, and that's why it's really the only homolog that is compatible with the soaking protocol. So we have also tried co-crystallization with the small molecule inhibitors of different chemical classes, and so far all of our attempts have failed with any of these other homologs. So it's somehow something about the macrocyclic nature of the natural products that makes it very easy to get good complex structures, but somehow doesn't work with the small molecules.
Mm-hmm. Yeah, good. And it's a general question for both of you. Anyone can answer. So, or both of you, both perspective, academic and industrial, would be better. So we know that from hit identification to the lead or the candidate profiling stage, it's a long journey, right? And there are in industry as well as in academia, there were a lot of efforts to reduce this kind of time period, right? So we would like to know from the industrial perspective first and followed by the academic one, how far we are at the moment, like using new technologies, for instance, the AI or any other technology which can reduce this time period, and we can get the antibiotics in market, because of the resistance is quite well developing. So we need to keep on producing as fast as we can. So we would like to know your perspective on that, starting with maybe industrial perspective from Patrizio.
Yeah, let me put it this way. Drug discovery is not easy, and drug discovery in antibiotics is certainly not easy. So the easy targets have been worked on for decades, and I think they're no longer available for new discoveries. And now we're really in the tough target business, and so AI, of course, promises a lot, but so far is not really delivering because, for instance, if you look at zosurabalpin, this is not something that AI would have predicted. Even when we knew the target, we asked AlphaFold about the binding mode of zosurabalpin to the LPS transporter, and it got all wrong. So, but yeah, we will never dismiss AI. We certainly keep our eyes open and see when it can contribute once the hype phase is over and we start to identify the useful parts of AI.
Yeah, great. And maybe Anna, your last take on it.
So I'm convinced that there's a lot of potential for AI to make a real difference in drug discovery. I would say it's still the really early days, which is of course not true for other fields. Think about ChatGTP. So I think the challenge now is that core computer scientists, not only bioinformaticians or chemoinformaticians, really discover their passion for drug discovery and start to learn the language, and then can together with experimentalists figure out what is needed to really make a difference. And I think a difference can be made at every single stage of the drug discovery and development process. In Saarbrücken, we are very fortunate that we have excellent world-leading computer scientists at the university and at two Max Planck institutes, and they have recently really joined forces and hopefully will be able to report on some nice findings very soon.
Great. Okay, with that, we have now come to the end of our question and answer session. So I would like to thank everybody for their questions and comments. And once more, thank you all, like Anna and Patrizio, for your time and really clear presentation, as well as the question and answer session afterwards. Now, Victor, I now hand over to you.
Great. Thank you, Ravi, for moderating today. And of course, thank you, Anna and Patrizio, for your great presentations. So you can already register for our next Revive webinars. On the 27th of February, we'll have one on methods for predicting human pharmacokinetics and dose of antimicrobials. And also on the 3rd of April, we'll have one webinar on using artificial intelligence and machine learning for antibiotic design. We are also conducting a survey to better understand how antimicrobial researchers have access to expert advice and what challenges experts might have in providing this advice. So please do help us by responding to the survey. You can access it through the link that I've just sent in the chat, and also by with your phone by accessing this QR code here. So that will be all from us. Thanks everyone for joining today and for contributing to this discussion. I hope you found the webinar interesting and useful and that you will join us again in future. Please do spread the word amongst your networks and encourage your colleagues to join us well next time. Thanks again and goodbye.